We are going to go through our drug binding interactions now. The purpose of this lecture is that you will be able to explain the difference between covalent and non-covalent bonds, at least be able to tell what kind of interaction a drug is going to have, identify the major types of covalent bonds that are currently in use in medicinal chemistry, be able to explain why each of the non-covalent bond has a unique distance requirement and bond energy. Also, looking at a drug molecule, analyze and identify the different types of drug interactions that are possible, and looking at specific amino acid functional groups present on biological targets, be capable of predicting the type of bonds that they will also be able to form. As we've mentioned a few times already in this lecture, in this class, covalent bonds are very important in the actual activity of the drug, but they're not used as much as non-covalent bonds. Okay, they are effective, but not used as much. So we have alkylation reactions that can take place, acylation, phosphorylation reactions, and also rearrangements. Non-covalent bondings usually occur when there is some type of electrostatic attraction. So let me go back 1 minute here and say in covalent bond you actually have sharing of electrons. So electrons from one molecule are going to be shared to the other one. So you have sharing of electrons. In non-covalent bonding you simply have attraction. Okay. So in ionic bonds you require compounds that have opposite charges. In dipole interactions, we have two kinds. We have ion dipole and dipole-dipole. Okay. Van der Waal and hydrophobic interaction, as the name indicates, happen when we have hydrophobic molecules. And finally, chelation and complexation involve the presence of positively charged compounds, usually metals, with negatively charged compounds, very likely to be either conjugate bases, you can also have negatively charged amino acids. All right, so let's move forward here. So here are some examples of covalent bonds. As I mentioned before, this is a strategy that is not commonly used. However, you have some very effective drugs that use that strategy. You have aspirin, which is an anti-inflammatory, also antipyretic, also has antithrombic activity. You have clopidogrel. Clopidogrel actually is going to use the rearrangement strategy. Chrombuthyl is an alkylating agent, and cefoxitin is an antibiotic that works by inhibiting cell wall synthesis, of bacteria. So the main concern with covalent bond is it has a really high bond strength, and so when something has such a high bond strength, the action of the drugs cannot be spontaneously reversed. So if you have, for instance, a interaction between ions, those interactions can be reversed. However, when you have a covalent bond that is formed between a drug and a specific receptor, you actually need to have specific catalytic processes or the formation of new proteins and enzymes for that effect to win off. And so let's take, for instance, the example of aspirin. Aspirin is a good drug. It's going to irreversibly bind the COX enzyme. And the COX enzyme is a cyclooxygenase enzyme. And so a lot of people are on low dose aspirin. Low dose aspirin. Okay, so it's going to bind irreversibly to that COX enzyme. For the duration, for the lifetime of the enzyme. So, in order to reverse the effect of aspirin, you're going to have to wait for new enzyme to be made. For new enzyme to be made, you're going to need some protein, some RNA, and for RNA to be made, you're going to need DNA. And so you see how these processes take time to happen. That's the reason why a lot of drugs that use the covalent binding interactions have a long duration of action, and when they have side effect, let's speculate that it affects the wrong receptor. When they have side effect, those side effects last really long and can be very detrimental. So that's one of the reasons why this strategy is not often used, but when it's used well, it can be very beneficial. So you want a drug that's using the covalent bond strategy to be specific to its target. Okay, you want it to be specific to its target. All right, so let's dig through the specific kind of bonds here. Now, the type of bond, covalent bonding that we're going to deal with right now is the alkylation. And the example that we will look at is chlorambucil. Okay. Now, alkylation occurs when you have a nucleophilic atom or functional group that's present on the receptor. So you have a nucleophilic atom or functional group present on a receptor, and you have an electrophilic group on the drug. Okay. So in the specific example here of chlorambucil is a drug that is used to treat Hodgkin's disease. Also used to treat non-Hodgkin's lymphoma. Okay, and some other neoplastic disorders. So chlorambucil is known to alkylate DNA. And the way it does this is the lone pair of nitrogen on the nitrogen is actually going to cause the release of the chloride, which is going to give you a very electrophilic position here. And then a nucleophilic micromolecule, such as the N7 of guanine, can actually attack at this position, causing the electrons to fall back on the nitrogen and leading to the formation of a covalent bond between that DNA and the chlorambucil. Okay, so when you have a nucleophilic attack of a lone pair of electrons that is present on the tertiary amine, you have a nucleophilic attack of the lone pair of electrons present on the tertiary amine on the beta carbon, it's going to cause the chloro group to be released. And once that chloro group is released, it leaves the highly electrophilic quaternary aziridium ion here. This is called the aziridium ion. Aziridium ion. Okay. And then in the second step of that reaction, a nucleophile such as the N7 of guanine is going to attack, causing formation of this covalent bond here. And so in this particular example, it's going to result in cross-linking of DNA, because not only do you have this happening in this, you also have a repeat of the same for this other beta carbon here. And you are basically going to end up with two different strands of two different nucleobases of DNA attacking the same chlorambucil molecule, leading to cross-linking of DNA. And since we're dealing with cancerous DNA, we can use that for our advantage. So it leads to an anti-cancer effect. Acylation is another process that we can take advantage of in drug design and development. So acylation is going to occur again when you have a nucleophile, you have a nucleophilic atom on the receptor, and then on the biological target you have either an ester, you have a lactone, you have anything that contains an acyl group, really. Anything that contains an acyl group. And so in the case of aspirin, for instance, aspirin is actually going to irreversibly bind to the cyclooxygenase enzyme. So the hydroxyl of the serine in cyclooxygenase is going to attack this electrophilic position, resulting in the acylation of the cyclooxygenase protein. Okay. Now, the reason why aspirin has activity is because the cyclooxygenase protein is involved in inflammation, in fever. And so when this enzyme is acylated, it loses its activity. So there are some other cyclooxygenase inhibitors, but aspirin is the only one that actually does it in a covalent manner. So there's other ways to block the cyclooxygenase enzyme as well. All right, so another example of acylation is seen with the beta-lactam antibiotics. So the beta-lactam antibiotics are actually responsible for irreversibly binding to a bacterial protein that is called transpeptidase. And transpeptidase is the bacterial enzyme that is responsible for cross-linking bacterial cell wall. So transpeptidase helps to build the bacterial cell wall. In a normal case, without the presence of the antibiotic, the transpeptidase enzyme is going to recognize the D-alanine sequence and bind, or rather attack the electrophilic position of the carbonyl and lead to an esterified intermediate in the first step. In the second step, there's going to be a cross-link that is formed and the transpeptidase is regenerated. In the presence of cefoxitin, however, because the cefoxitin and the other beta-lactam antibiotics actually mimic the D-alanine sequence, the transpeptidase is going to attack at this electrophilic position, so it's going to form a covalent bond with the beta-lactam antibiotic. However, there is no possibility of forming a cross-link, and the enzyme is covalently bound to the beta-lactam antibiotic. Now, the advantage of using the beta-lactams is the transpeptidase enzyme is specific to the bacteria. Okay, that's the reason why even with high concentrations of high doses of beta-lactams, you rarely see fatalities. I apologize for my terrible handwriting. I'm working on it. So that's another example of acylation reactions. Let's keep moving forward here. All right, phosphorylation reactions. Now, when it comes to phosphorylation, the type of covalent bond that are usually formed with organophosphates. And isofluorophate was once used as an ophthalmic agent. Okay. Today, most of the organophosphates that you see are used either as insecticides or as military nerve gases, and they're quite toxic because they irreversibly bind to acetylcholinesterase. Okay, so the mechanism of action is very similar. It's very similar to what we've seen before with the serines. So you have the serine hydroxyl here that's going to cause an initial phosphorylation. So it's going to attack at the phosphate here and lead to an initial phosphorylation of acetylcholinesterase. Now, at this point, this is still reversible. It is still reversible. Now, hydrolysis of either of the isopropyl group is then going to lead to a phosphorylated acetylcholinesterase that is not reversible. So at this point, at this stage, phosphorylation is not reversible. So the only way to reverse this phosphorylation is to use an antidote. So pralidoxim is the antidote that is currently used, and it is used because the oxime hydroxyl of pralidoxim actually has a higher affinity for the phosphate than the hydroxyl of the serine of acetylcholinesterase. So the oxime hydroxyl has a higher affinity and is going to displace acetylcholinesterase. It's going to displace acetylcholinesterase, leading to the regeneration of acetylcholinesterase. All right, let's quickly go and take a look at the rearrangement reactions. So rearrangement reactions are, again, another very effective drug in clopidogrel. And Rearrangement reactions basically cover drugs that have to be, they have to undergo molecular rearrangement, often through metabolic activation. Okay, so clopidogrel, for instance, is not the active compound. It has to go through oxidation to the 2-oxoclopidogrel, and then undergo tautomerization to the keto form, followed by hydrolysis to liberate the thiol moiety that's then going to be able to engage with the bond of the target receptor and form disulfide bonds. Okay, so the opening of this thiophene group here, the opening of the thiophene group here is going to liberate the active metabolite, and the thiol group is going to be able to form a disulfide bond with a specific receptor on platelets. So that's the mechanism of action of drugs that undergo rearrangement.